if we continue on this idea of the cancer model, you know, I talked about studying the breast cancer tumor metastasis, but we also know that one of the things that can really potentially cause inflammation in the liver is when things leak through the gut. So your gut, your intestine, all the blood from your intestines immediately goes to the liver. It regulates metabolism. But your gut's also filled with microbes. And the microbes, little pieces of them, leak across the gut wall all the time and interact with the immune system in liver. But if you get a terrible GI disease or maybe take some drug that changes the gut permeability, now all of a sudden the liver can see a lot more bacterial products that it's used to, and it can get inflamed. Maybe not to make you really sick, but a little bit sick, and that might be okay. But now if you're taking a drug or you have some other kind of stress, you may harm your liver. But also, if you've got a tumor cell sitting in the liver, it may now get stimulated by this inflammation to go from a quiescent state to a more active proliferative state. So how could we study that? Well, that brings us to our microphysiological systems program that's funded by DARPA and with input from the FDA and in collaboration with NIH. And there, DARPA had a vision that they wanted to have the ability to replicate many interacting representatives of organ systems on a platform that would let us look at crosstalk between these so-called microphysiological systems, part of the liver with the gut, with reproductive systems. And so they funded a substantial program, and I direct one at MIT with collaborators from Draper, University of Pittsburgh, Xioxal, a company, and Northeastern. And together, what we're trying, a big team at MIT, so we're building interacting organs, and one of the prominent ones we're working on right now, and we've just got our platforms all built to show, is interaction between a model of a gut that has immune cells and microbes interacting with the liver. So now, this program, we're not completely built it yet. We've got the platform built, we can have organ crosstalk. What we're in the process of doing is really building a very detailed model of the gut-liver interaction so that we can start to model not just lipopolysaccharides as our model inflammation. We can actually start to put real patient gut bacteria on a model of the human intestine and have it interact with the liver. So this is a very hard problem, as you can imagine, and it takes a kind of biology and biological engineering we have at MIT thinking about how do you construct this model so it has all the right parts. You can't make the whole gut, you can't even make the whole, you can't make the whole liver. What are the right parts to represent this kind of complex thing? That's where systems biology together with tissue engineering is so powerful. It tells us how to make the model complicated enough, but not so complicated it would be too expensive and unwieldy for people to use. That's what's really fun about this project. As an engineer, you're always looking for how can I do it the cheapest possible way and get the information I need, okay? Because you want to move on to some other exciting problem. You want to have everybody use it. So how did that project get started? Well, I think about it a lot because of breast cancer right now, but that project actually started from an industry collaboration many years ago. And in fact, this was a real problem that colleagues at Amgen had worked with in a rat model, and they said, you know, you just can't model this really. You can get some idea about it in mice and rodents, but what we really need is a human liver and the whole thing recapitulated. So they really stimulated us to think about this, and they've joined now our industry advisory board for our DARPA program and our NIH program, and now we have a collaboration with them. We communicate with them very frequently about this topic, and many of our other industry, particularly in pharma, these are the kinds of problems that are so really challenging to them, and so it's a very interesting two-way flow of information because they have the problem. We say, well, what if we did this? And then we go to their lab and we send people to their lab and we see how would you actually use it in Amgen or Pfizer or Novartis or Sanofi. You know, and these are all people we interact with regularly because you'd love to have something that's really perfect, you know, that's exactly a liver. But you know what? That's probably overkill because it'll be too expensive and hard to use, or maybe. So how can you make something that's good enough for what they need, easy for them to use, and captures all of that? And that's where really this marriage of tissue engineering and systems biology is so exciting, because engineers are all the time saying, how can I make something good enough to really do a great job, but that I don't over-design it, so I wasted all this money on features that people don't need. So we're in this very wonderful process right now of interacting with our industry partners to figure out what is it you really need, and let's build it to be that, because we can make it really complicated, but we also know that sometimes it's so complicated it's only artwork. And the analogy there is partly that you're going to have somebody pay millions and millions of dollars for something that's beautiful, but has sort of an intangible reward to you. Drug companies want tangible rewards. They have a pipeline, and the things we're doing add to their pipeline, add to the tools in their pipeline, and that's an important point. What we're building is not replacing animals right now. It's not replacing regular cell culture. It's adding another feature, and the way we like to try to explain it, it's bringing part of your clinical trial further up in the lab. Okay, so before you go into people, you're actually going to try to bring the people into the lab in a miniature form and be able to test things on them. So that's really the goal. How can we bring our ability to look at a patient population and bring the variability in that and so on, and have it be complicated enough to capture what we think is going to happen in the clinic, all the ways that the immune system can just really wreck you in drug development? How can we build that in, but not make it so complicated that it's going to be beyond the reach of you to actually use it in a meaningful way? Where have we gotten tremendous interest about our program? Certainly from people in the public are curious about it, but in industry, I think there really is a huge interest in how do we bridge the in vitro, in vivo correlations for efficacy and toxicity, and they now appreciate that it's very complicated by individual patient situations. Patients who already have some disease and now they're going to take another drug, or if they, as I mentioned earlier, get infection, how will the infection influence both the efficacy of the drug but the toxicity? How might underlying stressors like obesity influence the way a drug is efficacious or toxic in a patient? So we're realizing that there's many different facets of patients' states that we need to capture, and we need to do it in an efficient way because you can't do like 5 billion experiments. So one of the things that we view our role is for us to do a lot of experiments and figure out which ones are really the important ones to do. And along those lines, many of the investigators in my program who are in the area of systems biology have ongoing clinical projects where they're in the clinic involved with analyzing patient samples and working with drug companies to develop ways of personalizing, understanding the different patient subpopulations. So we have 5 faculty members very actively involved in this, including myself. I have a clinical project that I'll talk about in another clip, but this is a very important thing, that we have this direct continuum from patient studies where we go in the clinic and get the samples and analyze them all the way back to our platform, and we say, what did we learn from that? And more importantly, how can we take our partnerships with our industry partners where we're doing a systems biology analysis of patients and bring in their safety group or their early lead discovery group and put all of these people together in a huge program to really bring whole new ways of doing this personalized medicine from concept up to application in the clinic.